Hello, I would like to explain how we in the FDA's Center for Drug Evaluation and Research protect patients and consumers from drug quality risks. The reason we do this is because everyone deserves confidence in their next dose of medicine. Pharmaceutical quality assures the availability, safety, and efficacy of every dose. Let's take a closer look at the complementary tools that keep product quality risks from becoming patient risks. We'll start by looking at the tools applied by a manufacturer. It starts in the early life cycle of a drug product when a manufacturer is developing a drug. The manufacturer identifies the critical attributes of their drug and creates a manufacturing process and control strategy to produce it. Now, this is a very important point. One of the guiding principles of modern quality management is that quality must be designed into a product. Quality cannot be tested or inspected into a product after it is made. It is a result of the design of the product and its manufacturing process. This is a globally accepted concept in the ICH Q8 guidance on pharmaceutical development. These two scholars on the right, Deming and Duran, along with several others, were pioneers in the field of quality management. A tenet is that no amount of testing or inspection can overcome a poorly designed product or process. This is why so much of CEDAR's application assessment focuses on the applicant's design of the product and the process they will use to make it. In another step of a drug product's life cycle, a manufacturer establishes a specification, which lists the test and acceptance criteria that must be met before releasing their drug to the market. As part of this next step, all analytical methods used must be shown to be fit for their intended purpose. For example, a test to ensure the proper amount of active ingredient should be able to detect that ingredient among all other components of the drug product. If a manufacturer uses their own in-house method, it must be validated. If a manufacturer uses a United States Pharmacopoeia USP monograph method, it must be verified because it has already been validated by USP. For generic drug substances and products, many tests, analytical methods, and acceptance criteria are defined in USP monographs. USP is an independent organization that sets quality, purity, strength, and identity standards for medicines. Verification is easier. It is showing that the procedure can be used for its intended purpose under the conditions of use for a specific active ingredient or drug product. For example, a test must work given all components of the drug product. Validation is harder. A large series of studies must address a method's accuracy, precision, specificity, detection limit, quantitation limit, linearity, range, and robustness. And it is not only the finished product that is tested. A manufacturer also tests all components that will be used to manufacture the product, as well as intermediates formed during the manufacturing process. Before a drug product can be marketed in the US, a manufacturer seeks FDA approval by submitting an application showing they have designed a manufacturing process that reliably results in a product that passes all tests over its proposed shelf life. In an application, a manufacturer shows that they can make batches of product that pass all tests in the specification. For a generic drug, this testing is typically performed on batches at 1/10 scale, at minimum, of the proposed commercial process. Before an application is approved, the manufacturer also shows that the product passes all proposed tests over a defined period and when stored appropriately over its shelf life. If a product is approved before real-time shelf life testing can be completed, then the manufacturer provides additional testing data after approval to confirm the full shelf life. Note that for generics, there is often less stability risk due to knowledge of the innovator's drug's stability profile. Once FDA approved, a manufacturer scales up and validates the process and tests each batch before release. Since the application batches for generic drugs are often made at smaller scale, a manufacturer may need to scale up their process after approval and validate it. Scale up may involve adjustments to the manufacturing process and the use of larger equipment. This larger scale process must be validated. Process validation was founded on the acknowledgement that one-time testing of a final drug product is not enough to assure patient safety. Validation means that the process is defined, the process is evaluated, and the process remains under control. Note that unlike traditional batch processes, continuous manufacturing processes are designed to scale up by increasing runtime, so there is often less scale-up burden after approval. A manufacturer tests every batch they make based on the approved release specification, and only passing batches can be released to market. It is important to note that product testing can be destructive, so product removed from a batch for testing might not make it to the market. A manufacturer samples from a batch using a sampling plan that provides appropriate confidence for assuring batch quality. Release testing isn't the only product testing that a manufacturer performs over a drug product's life cycle. A manufacturer performs ongoing stability testing and maintains the integrity of the data. A manufacturer must manage the ongoing release and stability testing and ensure the completeness, consistency, and accuracy of data for all regulatory records. This includes data related to the product and manufacturing process. Even after establishing an appropriate shelf life prior to approval, a drug is approved with a stability protocol that defines the ongoing stability testing that will be conducted over its entire life cycle. The data generated by this testing is one focus of FDA inspection. Now, let's take a closer look at the tools CEDAR applies to protect patients from quality risks. To facilitate drug development early in the life cycle, CEDAR releases guidance on product development and establishes quality standards, including at times the test and criteria for a product's release. It is important to note that manufacturers do not typically make up all their own tests and criteria. CEDAR releases guidance to address tests and criteria needed for drug development and release. For example, there are the International ICH Q3 guidelines on impurities and numerous drug-specific guidance documents on the demonstration of equivalence for generic drugs. CEDAR also works with standard-setting organizations, most often USP, to establish the minimum tests and criteria in general chapters, which can apply to many drugs, and monographs, which apply to one specific drug. After receiving an application, CEDAR assesses the application for product and process quality and conducts an inspection if one is warranted based on risk. At this point, let's remember the lesson of the quality management scholars. Nothing is more important than showing that the product and the process were designed to generate a quality product, one that is safe, effective, and free of contamination and defects in every dose. If the application is for a generic drug, then thorough tests must show it is a therapeutic equivalent to the innovator drug, meaning that it can be expected to have equal effect and no difference when substituted for the brand name product. To be a therapeutic equivalent, it must be, among other things, a pharmaceutical equivalent and bioequivalent. Pharmaceutical equivalent means having the same amount of the same active ingredient, dosage form, route of administration, etc. Bioequivalent means performing in the same manner. At this point, a pre-approval inspection may be conducted if warranted based on risk and/or history to in part confirm the data, including testing data, submitted in an application. Important to note that for changes made after approval, manufacturers also must test the product and demonstrate it continues to meet specification criteria before implementing the change. After a product is approved, CEDAR uses surveillance inspections and remote regulatory assessments. After a product has been approved, surveillance inspections are one way that CEDAR monitors the manufacturing process and quality of products on the market. Note that all manufacturing sites are subject to routine surveillance activities. CEDAR's site selection model prioritizes sites for surveillance inspection based on risk. A surveillance inspection or remote regulatory assessment covers data integrity and manufacturing practices for all products and processes at a site. Once a drug product is on the market, CEDAR conducts data-driven market surveillance. CEDAR's surveillance program uses analytics in a variety of quality data sources such as post-market quality reports, inspection reports, and application data to find potential quality signals. These data sources inform the site selection model that prioritizes surveillance inspections and the strategy for product sampling and testing. In fact, CEDAR developed and deployed a risk-based statistical signal detection algorithm to inform sampling assignments. This algorithm prioritizes drug products and targets the types of tests that could uncover quality issues. The final tool we'll cover is conducting risk-based product sampling and testing. Put together with the other regulatory tools and the tools of manufacturers, the residual risk to patients can be minimized. It is important to understand that although residual risk can be minimized, it cannot ever go to zero. This is why it is important for both manufacturers and regulators to play their parts in keeping patients safe. Let's take a closer look at product sampling and testing. CEDAR uses a combination of an expertise-based approach and an analytics-based approach to identify problems that warrant testing. I've already discussed the analytics approach, so let's cover the expertise-based approach. Experts in the quality assessment, surveillance, compliance, and inspection disciplines are solicited for their recommendations. It is important to understand that in some situations, a suspected public health hazard can be resolved faster with another FDA action, such as requesting recalls or issuing warning letters. Sampling and testing can be assigned at any point in the year, so it can be responsive to risk, workload, and developing issues. This data-driven approach means that there is not typically unbiased or randomized testing, as this has not proven to be a good use of testing resources in the past. Typically, a product is tested if it has been signaled by experts or algorithms for surveillance testing. Now let's discuss how sampling and testing works. CEDAR determines the amount of product to sample based on the type of testing to be conducted, number of tests needed, the size of the lot or batch under investigation, and the amount available. It is important to note that no single test can be used for all quality attributes, so tests typically focus on the attributes with suspected risks. Further, it is very important that the amount of product sampled for testing by CEDAR should not significantly impact the amount of product available to patients and consumers. Sampling removes a portion of the product from the market, and it must be done carefully as to not exacerbate drug shortage risk. FDA collects enough sample needed to verify or validate the method, perform the testing, perform confirmatory testing, and retain product should results be contested. With more than 140,000 drug products in CEDAR, no sampling strategy can be representative of the entire market. At times, CEDAR conducts proactive studies that use statistical sampling to make inferences about a targeted population of interest while quantifying the uncertainty due to sampling. For example, this strategy was used recently to sample hand sanitizer manufacturers. Testing the quality of product for the market is best done by a manufacturer running a process designed with appropriate controls, including release testing with an appropriate sampling plan. A final note and a look forward. Additional information, not necessarily from additional product testing, but rather from a manufacturer's use of additional tools such as quality metrics and quality management maturity assessments, can further reduce supply chain risk. Quality metrics can help monitor a manufacturer's sustained performance between points in time and help identify opportunities for improving the manufacturing process. Quality management maturity assessments will be voluntary and conducted at points in time to encourage manufacturers to implement robust quality management practices. Beyond these developing programs and far into the future, we will continue our efforts to better assure that quality medicines are available to the American public.